Thank you, Michel, and organizer, for the invitation. It's a pleasure being here. And as Michel said, I will speak today about my research in bioinformatics and in cheminformatics. Um, and even if the topic here is really focused about trying to accelerate in general virtual screening method for RNA, the underlying message I would try to bring is more about how to have better representation of RNA to take advantage of the fact that RNA are folding, meaning the structure, to accelerate and build a custom machine learning framework for RNAs. All right, so the justification, the motivation, where I will start with today is really try to show you how we can increase the accuracy eventually, but the speed of predicting small molecule binding to RNAs. And of course, part of the talk is how to make better use of machine learning techniques out there and make it efficient. But the real message I would want to bring here is that if we try to have a better model of representing the RNA itself and the weight fold, we can, by better understanding the RNA itself, use it to use basic machine learning techniques to accelerate all these tasks. And I hope that the prediction of small molecule binding RNA will be a good example. So first of all, why do we care about predicting small molecule to RNA? Is just a bit of perspective. This is a very nice review by Kevin Weeks and co-worker. The peer, like 6 years ago now, that reintroduced very well the motivation of all this. If you look at the drugs on the market, like it's they're basically all targeting proteins. But now, if you look at proteins, basically represent 1.5% of the genomes, and the druggable one, the very fraction that you can use, is even smaller, up to a very 0.05% are the target we're using for existing drugs. So it's very tiny. Now, by contrast, so there's no drug really used for targeting RNAs, but RNAs can encoding about, we estimate up to 70% of the genomes. It means there's a lot of target out there that we don't use, and we have no clue how to access it. So if we want to address this question, we cannot rely on the technology we had up to now because it's too slow. We need to develop better techniques to go faster and try to eventually bring the field to the point where we can use the well-established techniques to help us achieving that vision. So all the work we're doing there aim to achieve this. So the classical pipeline I'm interested in is what we call the structure-based RNA visual screening. It's a very arbitrary view I'm presenting here because potentially not everyone has the same representation, but eventually what you want is once you have a structure here, okay, found it here. You'd want to take some libraries of small molecular compound and try to dock them on the 3D structures to see if it's potentially a good binder. And docking one molecule is not that very expensive. It's sort of computational simulations. But the problem is you want to screen many of them. And so that takes a lot of time. And the problem we have with RNA as well, if you look at the PDB here, the protein data bank, so basically the repository of crystal structure, there's very few of them. There's order of magnitude less structure known of RNAs than protein, despite the fact that it's much more a potential target of RNAs encoded in our genomes. So if at some point what you want to do is to be able to have a computational pipeline to predict the structure, and typically it goes from this way. So you start from a sequence, try to predict the secondary structure. We have good tools for doing that, like the RNA fold or RNA structure method software. that have predicting relatively well the Watson-Crick base pair and Wobble into the RNA, so the secondary structure, and that can be applied massively. And then after you have a small of a gap, if you don't try to move from a secondary structure to a tertiary structure, that would be what you would need to apply the classical docking system and the simulation. Then it starts to be like the twilight zone nowadays, right? You don't have, you have some method out there, but none of them works very well with, at best, resolution of a 5 angstrom RMHD, which is good, but not enough if you want to do docking. Because eventually, even if you have these 3D structures, you have to keep in mind that a small variation in the change, some very small inaccuracies in the pocket where the binding should occur, that wouldn't even be noticeable when you try to build a full RNA, just doesn't, will invalidate the docking simulation. So we're not there. That's the bottom line. So what I would try to do today is like this challenge of trying to go from a sequence up to the tertiary structure is not close to be solved anytime soon. Well, I mean, when I say anytime soon, don't quote me here, because maybe in a couple of years we may manage to do it, but at least not as of today. The idea we're trying to push here is like, well, there's many things that goes wrong with RNA. First, we don't have many structures, but also they're a bit flexible. So I mean that many methods that we use classically for docking the molecule don't work well. So what we want to do is just to skip that step. We're trying to see if we can try to take representation of the molecule that is coarse grain at that lower degree of resolution and try to apply techniques to determine, to dock the molecules or to find potential binders that are more efficient. Perhaps not the same accuracy, but at least I won't require these 3D structures. And if we do that, the big advantage for us is because the representation is discrete. It's is simpler. We have better hopes to be able to have efficient mathematical model and machine learning approaches for solving this issue and answering the question. But of course, that also requires that the representation of the molecule you want to have include enough biological information to say something meaningful. So there is this threshold, this balance between both that we're looking at. So a lot of work has been done at the secondary structure level. And this is one of the key feature of RNA is like, we know that RNA folds quickly, first create the secondary structures, and then form the 3D structure. And usually the secondary structure is basically the set of all Watson-Crick and Wobble base pair that you will find in RNA that is miraculously form often a planar structures. I mean, sometimes you have long range, but I won't discuss about this. And it's a very nice representation, very well conserved. So structure is more concerned with sequence and RNA, and for which we have many tools that have been developed for predicting the structure, for identifying eventually the giving to the function, but also to find some molecular interactions. So a lot of work has been done also for the group, for instance, Matthew Disney and the Scripps Institute with InfoRNA, that is about using the secondary structure with libraries of motifs they found to predict some small molecule binding it. But it's not limited to that. You also have many methods based on secondary structure for predicting interaction with other RNAs or for LBPs as well. So the problem we have is like secondary structure is good, but can we go beyond that? Are there intermediate level of representation that contain more biological meaningful information that is still represented as a graph so that we can use in computational models efficiently? And that will help us to basically build efficient models for screening these RNAs. And there's a very important notion. Basically, there's one slide that you have to remember from my talk here. Maybe is that one, and it's not my contribution, but it's a contribution from Eric Westhoff and Leontis, who basically introduced this vocabulary, if you want, this classification of all base pair occurring in RNA. So you all know the Watson-Crick base pair, which is a classical one, and the Wobble appearing between AU, GC, and the GU. But the truth is that RNA is have a complex side chain, and they can interact in 12 different ways. So typically, the base of RNA here has 3 edges that we call the Hookstein, the Watson-Crick, and the sugar edge. And the very well-known Watson-Crick interaction that we see in the RNA, in the secondary structure of RNAs, is often the one happening between the Watson-Crick edge in a cis conformation. But all nucleotides could eventually interact with any of these edges in a cis or trans conformation. And that makes you 12 different types of interaction possible. So if you start annotating this RNA structure with this base pair notation, what you will see is a much more complex network. And often this interaction, this non-canonical interaction, as I'm calling it here, and we usually call here, are often located in the hairpins, the loops in general, the multi-loop, the bulges, the internal loops. And that give you really very different perspective about how you perceive RNA structures and this graph. Often when you look at RNA secondary structure, you look at like a multi-loop or an hairpin, and you have the feeling that these regions are just very flexible, they're going a bit everywhere and not very important. But the truth is like this is exactly where things are interesting, because this is where you have complex interaction networks happening, and that basically shape the 3D structure of your full RNA. And one of the work of that's been shown by many different groups, there was so that this network of non-canonical interactions is also a very strong signature for the 3D structures of these motifs. So when you have some specific networks of non-canonical interactions, and when I say non-canonical, that also include canonical, so it's all 12 interactions together, you have a very strong association between these motifs and the 3D structures of the fragment it represents. And this is what we want to use here to develop our method. Can we use this expanded representation of RNA structures to build method for predicting the function, in the case, the binding of small molecule to RNA? So the main work I will discuss today is this project that we started with Carlos Oliver, did his PhD with me, but now moved to the Max Planck, and I think come back to US soon. It's called RNA Migos, and it's really work driven by Carlos and Vincent Mallet, who is now at LMI in Paris. And the idea was can we take advantage of this extended representation of RNA secondary structure to make better small molecule binding predictions for RNAs. So the idea of the project was really like once you start with a 3D structure. So I'm assuming here I'm starting to work. I know the binding site, so it's really the same problem as the docking. You have 3D structures. You know where the molecule will bind. You have the binding site, and try to predict if this molecule can bind to or where it fits to this pocket. So what we're doing here, like we're starting from the once we identify this binding site, we're annotating it with this non-canonical representation of all base pair interaction occurring there. That basically, and I don't know why there's some edges disappear here, but technically there's supposed to be a line, the backbone here, like it's but it gives us this graph annotated with every node representing here a nucleotide, and the edges in between represent one of the 12 type of non-canonical base pair interaction possible. And once you have this graph here, we want to be able to predict one molecule that would bind to it. So that's a bit of generative approach. the way we set up the problem. The molecule here that we try to predict actually is not exactly molecule. We try to predict the fingerprint. So that's the way we're representing the molecules. Basically, you look at the molecular structure of a molecule. You look at all different patterns, chemical bond that can be there, and you have this giant binary vector that represents all these features, and that's basically the representation we're using here. You have more advanced ones, like the smiles, the selfies, that can be done also. I mean, I'm happy to discuss it, but it's not what we've been using here so far. Why are we using this binary vector representation? It's because it's very easy to deal with. It's that we have a lot of metrics also to compare to different molecules by what's called a Tanimoto coefficient. It's basically just you try to make the product between two. And basically that's very convenient to use, and it still works to give, has been shown to give good performances. So the RNA Migos pipeline that we developed is really the following. So we start to have, first we take as an input the representation of the binding site using this Leontis-Vestov representation, so the non-canonical representation. And we basically use, we encode this, we have an encoder that basically we embed this network into a latent space in which we try to learn the target here that is associated, that could fit this potential binding site. And the RNA Migos pipeline, the first version, because I come to the second one later, is really, I give you this input network, and it would generate here a fingerprint that represents the potential molecule that binds to it. So the big issue we had, though, is really that the amount of data available here to play with. So there was about 3,000 non-redundant structure, not created by us, but it was there. And we had to remove the ions because there's no way to do anything with it anyway. There's sort of quality variation anyway. So at the end, we just end up by having 200 unique ligand and 700 binding site, which is relatively small, but that's the best we can do. And I'm not aware about any better data set at this point from crystal structure. So what was interesting for us, like when we try to use and implement this framework, try to learn this data, we try to evaluate the performance, try to answer the question, does this representation helps and boost the performances for predicting the binding of small molecules? So actually, you have to read this, well, I can guess here, from the right to the left. And what we've been doing is just try to have we try to predict, we have a set of decoys and try to see if among the molecule, the fingerprint generated by RNA Migos, if we can retrieve the one that was identified initially as a native. So in other words, basically you just want to see the rank of the native, which is basically the higher the better. I mean, like you can guess here. And initially we have 3 tests, basically just baseline. We just randomize swapping the labels to basically see the performance of the model when there's no signal in the data. We see it's around the 50%, so it was kind of good. The interesting thing starts here. Basically, when you use just the sequence, so just the secondary structures, we see that you have something that is barely better than just random. When you lose the secondary structure, you have a slight bump, but it's still minor. When you use here, after you augment the data by having this non-canonical representation, we see a clear bump into the performances of the model. So in other words, of course, we have a richer model, but the bump is still significant, and we have a much better performances retrieving the data. We compare also the model with another one called InfoRNA, which is developed by the lab of Matthew Disney at the Scripps Institute, which is basically only based on secondary structure. And unlike a model, it doesn't really include this potentially bringing together 2 different motifs. So it's a very unfair comparison to InfoRNA, because here we're dealing with much complex binding site. But just to be sure that the result we had before was consistent, with the one we just had in a previous experiment I showed to you. We also see here a gain in terms of including non-canonical interaction into the model. However, there's still many problems in this. One of them is that it works well on some classes of molecules. So some the aminoglycosides, for instance, are well, the performance are okay. But there are some classes, some proteins depend. On the fact of working on proteins better, I can always have to say it at some point. But some molecule basically more exotic, which the performances of the framework doesn't work well. One reason we have for that potentially is because, as I showed to you earlier, we really lack of data to make a good training. So eventually, we show the model is valuable. There is something to do here, but we really lack the training data to make it widely applicable and usable in real settings, basically. Specifically because, in addition, we have this generative task where we wanted to generate new molecules. And part of the story too is that if you look at the compound, the ligand identified in the PDB structure, in the crystal structures, often turns out to be not really the one that are drug-like molecules, not really the one that you want to develop as a drug. So there's discrepancy here about the one we know how to predict well and the one who would like to be able to predict because they are the ones that have the pharmaceutic interest. So the story could end here, but of course there's a follow-up to this. We decide to try to address this problem of the lack of training data and try to use have a more practical version of RNA-MIGOS. And this is where with RNA-MIGOS 2, which is the new framework, we try to use here data augmentation. The idea being that, okay, we don't have, we acknowledge we don't have enough data, but what we can do, we have some docking software that have reasonable performances, accuracies. And what we can do is to track 3D structures we know, we can take ligand we know, we can simulate the binding and use this data, even if it's very noisy, it's not perfect, we can still use it to train and try to learn from this data. And this is typically what we did here. So basically we keep our original data set. We also, and we run this massive experiment where we docked, we had 1 million, 1.3, actually I don't know what's 1.2, but it's 1.3 million docking experiment that we run to be able to see if we can learn something from this experiment. And then after we'll be able to train the model. But in addition to that, what we also do is try to have a task that is more focused. Instead of trying to generate new molecules, we try to focus on just docking the molecules. Trying to say, okay, I give you the molecule, I give you the binding site, and try to predict the affinity score, the binding affinity. And this is what the first version that we call AF here does. And we have an even simpler version that we call compat, which just tells us if there is a compatibility. It's basically a binary classifier, yes or no. So it should be that we try to simplify the model, and at least the answer it gives to the simplest expression. So the idea, the way it works is like for the molecules and RNA, we have two encoders. Basically that to resolve and have to decide to use either the affinity model or the compatibility model. And in a practical case, the way you using it is like once you have a pocket, basically what's the binding site you're targeting, and you take the libraries of ligand, and for each pocket and each pair of ligand and pocket, RNA-MIGOS will return to you predicted binding affinity. So what we show here, the first thing is like, well, the first validation, like we use RDoc as to simulate all the docking and try to find to see the relation between the value returned by RDoc and the value returned by RNA-MIGOS. And here you have a correlation that is not perfect, of course, but still quite acceptable and works well. And then after we move to a virtual settings model where we have a library of molecule, you're trying to dock all your molecules and see how much they enrich, what the rank of the ligand and the good binders to your target. And what we see, of course, like when you use just the compat and the affinity model, so two basic models, we're a bit lower than RDoc, but we're not that far. So that's relatively encouraging. I mean, that we really capture what RDoc is doing, massively speaking here. What is even more encouraging is like when we start to mix the two models and try to take an average normalized score of the compat and the affinity model, we've been able to boost better the performances. And we can even include that with including afterward, and we'll speak a bit more about that next slide, 2 slides, sorry, by incorporating RDoc score to really have good performances of the virtual screening settings. But before moving to a more practical application, what was also important for us to check is like how generalizable the methods are. And so one very quick check we did here, we look at all the binding site and the ligand that we had in our data set. And we look at the distance from this target site from the closest example inside the training set. And what we can see here is like basically that distributes quite well. So here you have the distance from the closest example in the training set. And if you're close or if you're very far, basically the performance doesn't seem too much affected, at least for the binding site. I mean, it's not perfect, of course, but that was relatively reassuring, saying that we're capturing something that is that can be generalized and used on a new target eventually. But now that's potentially the most practical use of the platform we developed here. I don't know why the image is not very clear. It's a bit blurry. It should be okay on my screen, but anyway. We try to combine both approaches because ultimately what we want to do is to give you the best confidence into the candidate we're generating. And in practice, if this screening was happening, it's like you have millions of molecules. You can only test very few of them, maybe maximum 1,000 of them that you have picked carefully. And you have to trust, you have to use this docking tool because that's the one we trust, that's the one that have the most interpretable result here. So the way we implemented the method is that first we use RNA to basically screen massively a very large database. We do basically filter it and extract the best candidate ligand. That after that, the most promising one were docked with here RDoc, any docking software that you like, to have more interpretable values to rescore if you want the ligand with a value written by these docking simulation tools, something that chemists are more used to deal with and can better interpret the result and the meaning of the binding. And what we basically show here is that in these screenings we have almost for all pockets here we increasing we have a gain in terms of accuracy, and more importantly we're really boosting drastically the time of the experiment. We have a 1 order of magnitude better virtual screening speed than if you just use the classical docking techniques. And what it means is like not that you run your computer less, but actually you'll be able to screen much larger libraries. This is what is interesting. Time is not about the time of the completion experiment, but just how large is the library you can screen. Finally, the last test we did, more like a real test scenario that we try to apply our method to, is this Robin dataset. So Robin dataset is a very nice assay that was published last year ago. That is published. Basically, you have 4 RNA target that are the ribose switches that you see here at the top, and the test that in vitro assay when basically they test the binding affinities for 20,000 compounds. And long story short, we try RNA-migos on that, and we saw significant enrichment of enrichment factors of a method that is even better than the one you would obtain with docking method. So we're very confident that we're capturing here something in that is valuable for screening the method. We also look a bit, if you look at this projection here, is basically how the different prediction are done. Well, we try to see basically that distribute well. There are some regions we miss, but I mean, this is not a hard fact, but there are some regions, for instance, I cannot remember, that we miss, but sometimes it's because some binders identified by Robin add some sulfur atom, which is basically not a drug-like property that you would like to have. So eventually what we hope it shows, and the thing we're trying to investigate, is like it's not just predicting binding affinities, but binding affinities and at the same time identifying molecules that have a good drug-like properties to push further in a pipeline. So I skip that one. So I'm getting close to the end. I want to keep a bit of time for the question. So, but what I want to mention here is like, and this is really the work of Carlos and Vincent, which is the two. I mean, they were graduate students when they start now. That they're both PIs, so I mean, it doesn't make me any younger. But they really develop all these libraries while in the lab and pushing it now. It's called RNA glib, which is basically a Python framework that helps you to better taking advantage of and better manipulate these RNA extended secondary structures for machine learning applications. So we use RNA glib here for RNA-migos, but potentially there's many other use that can be done. What it includes, like it has a created list of all PDB structure there, having also non-redundant libraries inside. They're all annotated using this non-canonical base pairing interactions. And there's many utilities here helping you to manipulate easily these data for PyTorch or DGL applications. So it's really a library that they're actively working on to basically make these approaches model accessible to anyone to develop your own favorite RNA bioinformatics application. I show it to you today as an example of binding of small molecules, but it's not limited to that. So I won't really have time to speak about it, but just saying that what I showed to you here is about the RNA-migos. Like once we have these 2.5D structures or extended secondary structures, try to improve the docking. But the other part of the problem is like, how can we go from the sequence and directly predict these 2.5D structures? And hopefully that's a bit easier than trying to predict the 3D structures, which let us give us a bit of hope that we'll be able to reach that goal relatively soon, if not me or other groups. But I just want to point out that the work of Roman Sarrazin-Gendron here, we developed this base pairing software, whose goal is precisely to, starting from a sequence, you want to predict occurrences of these different motifs into the RNA, and that combined with the secondary structures could give you this extended secondary structure data that we need to apply the RNA-migos pipeline. So I won't go through all the slides explaining all that work. I would just close my talk now, saying that, again, my message, I mean, I'm redundant here, but I think that's really what I want to push, is that all the machine learning method we apply there's nothing fancy. It's all very basic stuff. It works because we use the good representation, and it is really what matters here for RNA. There's many methods out there. We don't have to take just what works with protein and apply it to RNA. RNA has spatial molecules. I mean, I'm speaking as a structural biologist here. So they're very specific way of folding and way of interacting and creating some different type of interactions. And if we have models that capture well this information and we understand the molecules, then we have potentially a lot of different opportunities at developing very efficient models with very little effort. And this is what the RNA glib library here that we have is trying to push it. And the long-term vision of this is not just to predict the molecule, but our real vision is we try to see, once you have this sequence, can you start predicting the 2.5D structures, use it to make better prediction of the ligand, or better docking method, but eventually, after that's a long term, try to build also these 3D structures to try to better understand the synergy between both, and try to apply in the docking tools to understand how the 3D structure and the ligand fit together, and developing much more RNA-centric virtual screening tools. And so I will close here my talk by acknowledging all my students and collaborators on this project. I probably forgot a lot of people. I just want to shoot out for another upcoming talks of Jay, with presenting a very cool application of nanopore. Not yet fully a 2.5D representation, but hopefully very soon. So thanks again.
We have an online question. The question is, given that fingerprints, MAX, CFP4, etc., cannot be reversed back to a unique molecule, the predicted molecules are limited by the database of molecules used for Tanimoto comparison with a fingerprint. For the Amigos, RNA Amigos web server, what background database of ligands are currently being used?
So, well, I think for the web server, I think we decommissioned, because now we're moving more toward that. We realized the web server has some maintaining everything. So we're moving more to this GitHub repository that I think I put the things here, which is a more up-to-date, so RNA Amigos 2. So we use first, in the first version, the RNA Amigos 1, it was only ligand that we get from the X-ray structures, from the PDB database. Now we also included the Campbell database here to try to expand data sets. So, but eventually now the RNA Amigos 2, which is the model we're using now, you can use any drug-like molecule that you like, and we'll build this fingerprint representation, and we use it in the framework. So we're not really limited by database, but of course things closer to what is in Campbell and according to the drug-like property that, as we define them, would be more appropriate for the using. So I think that's, you have to look in a bioarchive preprint. You have the list of the properties. I don't remember them here, but it's.
There's the next part. So are there any future versions planned for direct SMILES predictions instead of to overcome known issues with Tanimoto's similarity metric?
Yeah, absolutely. I mean, we try to work now. There's 3 things we're working on. And first is indeed the goal was never to stick to fingerprints. It just was easier for us. It was simpler. But SMILES, which trying to work on SMILES, and selfies, even developing our own version of SMILES to be a bit more expressivities, is one thing we're moving on. We're trying to eventually go back to the generative model. I don't believe too much into it right now, but we play potentially with it. And something missing in the framework as well is the binding site predictions, which is one of the big question in the field now. So if I give you a structure, can it predict where it would bind? Some work have been proposed recently in the last month. I didn't read all of them, but it's a very exciting direction. We try to look at it as well.
Very nice talk. So sort of in line with the questions that you just had, I think Anne-Marie Pyle from Yale, they had another type of representation for structure for RNA, right, which is like a language for defining the backbones and the various angles. So how does that compare? Did you try to use this versus this? I'm just curious.
No, we didn't try. I mean, it's because, it's probably more historically, because, it was doing well, Eric Westhoff and on this representation. It's a very simple one. It's very almost straightforward extension. I think the representation again is a bit more advanced. That probably include more sophisticated approach to deal with with it. It's definitely something interesting, but we haven't touched that. But I'm not saying that's the ultimate approach. I'm just proposing one and just make the case that it's very good to work on the representation of RNA structure, because this is what will boost ultimately all this AI wave. It's not necessarily in the method, it's really in the model we're using.
A last short question.
I was just wondering how you mentioned that you're doing the representation, but a key to representation of first having the structure. And how do you deal with the fact that many of these programs like RNA fold and RNA structures give many structures and some of them are equal in energy? How do you choose, or how do you create a representation that represents all of them together? Or is that something you don't consider?
Well, I mean, here what I showed you for RNA amigos, we're starting with the RNA structure, a 3D structure. So we don't have this case because we have, once you have the 3D structures, we can and you have directly access the non-canonical base pair interactions. We use the Fred database from Craig Zebel at BGSU. So we have this information. Eventually, what I was discussing, sorry, I had to go very fast toward the end, but indeed, when you look at all this ensemble prediction that we call from that typically do in the field of prediction, you will have perhaps to deal with more probabilities of binding between different factors. We haven't played with this, but it's eventually something that we need to annotate, like saying there's a probability of X having a non-canonical interaction of this type here, and see how the method reacts to this type of information. But we have not investigated that yet.
We have to move